Welcome back to the Easy VC Brief. I'm your host, Max von Polnitz, and this is your weekly espresso shot to deep tech venture capital news. This week, I'm coming to you post live from Syn Bio Beta 2026 in San Jose. For those that haven't heard of it, Syn Bio Beta is the Super Bowl of synthetic biology. Every May, the engineers, the founders, the incumbents, and of course the VCs descend on the San Jose Convention Center to talk about one thing: using biology as a technology platform. I've been coming for a few years now, and every year I walk out thinking the same thing. We are genuinely living through some of the most important moments in the history of life sciences. This year was no different. 3 stories today, all from the show floor, all connected by one thread. We are learning to read biology like code. And some very well-funded people are betting that whoever builds the best reader wins. Story 1. Pumpkinseed BIO just raised $20 million, and yes, that's a real company name. Named after the most colorful freshwater fish in North America, apparently the shimmer of the fish's scales inspired the shimmer of their silicone chips. Pumpkinseed is a Stanford spinout co-founded by Professor Jen Dione, who I watched speak at the conference this week. The company just closed a Series A co-led by NFX and Future Ventures. So what do they actually do? Think of the proteome, all the proteins in your body, as the operating system that runs you. Genomics gave us the source code, but proteins are what actually executes the program. The problem is most of those proteins have never been read. They're invisible to current tools, either too rare, too modified, or too novel to show up in traditional mass spectrometry approaches that rely on reference catalogs. Pumpkinseed built something called Decipher, a nanophotonic chip platform that sculpts light at the molecular scale to read individual proteins, amino acid by amino acid, without needing a reference. Their chip packs over 100 million sensors per square centimeter, and each sensor concentrates light into a volume smaller than a single protein. I know that's a lot of deep tech talk, but think about it. Think of it this way. Existing tools can only identify a song if it's already in the database, a la Shazam. Remember that app? Pumpkinseed's chip can transcribe a song it's never heard before, note by note, and hand you the sheet of music. They're already working with Genentech on oncology applications. This one is definitely one worth watching. Story 2. Paradromics is putting computers in brains, and the FDA just said yes. Matt Engel, founder and CEO of Paradromics, took the main stage and delivered what I would describe as the most quietly confident pitch I heard. No hype, just here's the problem, here's the device, here's the timeline. Here's the deal, though. Millions of people globally are locked in, paralyzed, unable to speak, unable to communicate because their brain can't talk to their body anymore. ALS, stroke, spinal cord injury. The brain is still perfectly fine. The connection is just broken. Paradromics built the Connexus BCI, a fully implantable brain-computer interface, BCI, that records directly from individual neurons at the highest data rate of any device in its class. The FDA granted them investigational device exemption approval late last year, making them the first company cleared for fully implantable BCI specifically targeting speech restoration. Their Connect One clinical study is now enrolling 2 patients, we heard, at sites in UC Davis or Harvard's Mass General. He also mentioned a 4-year path to regulatory approval, which I think is highly ambitious, but the clinical logic is really sound. Start narrow. People with severe motor impairment who have essentially no other options, demonstrate safety and efficacy, and then expand. Wheelchair users today, eventually applications in chronic pain, mood disorders, and even consumer interfaces. This is one of those technologies that starts as a medical device and ends up being a platform. As a venture capitalist, it's a platform worth diving into. Check them out. Story 3. Darpa is funding a moonshot to read the dark proteome. I'll explain that. And it might be the most important biology program you never heard of. Quick primer on Darpa for anyone who's new here. The Defense Advanced Research Projects Agency, it's a mouthful, is basically the US government's mad scientist division. They don't fund incremental research. They fund things that seem impossible until they aren't. The internet, GPS, stealth aircraft. When DARPA puts money behind something, it's worth paying a bit of attention. As Syn Bio Beta this week, I watched a fireside chat that featured Leroy Hood, one of the most decorated scientists alive, the guy who co-invented the automated DNA sequencer, alongside Michael Corus, a director at Darpa's Biological Technologies Office. The topic, protein dark matter. Stay with me here. Here's the setup. Genomics gave us a map of the genome, but the proteome, all the proteins that genes actually produce, is still largely illegible. The vast majority of proteins in any given biological sample are uncharacterized, unknown in function, invisible to current tools, and completely untapped. Leroy Hood calls this the dark proteome, and he's now building a new peptide drug company on the thesis that next generation of medicine lives inside it. DARPA apparently agrees. They just launched a program called PROS, Protein Sequencing, with an audacious goal: build systems that can read any protein de novo without reference sequences at greater than 99% accuracy. And here's the thread that ties the whole episode together. Pumpkinseed's Decipher platform is exactly the type of technology DARPA's PROS program is trying to catalyze. Hood is defining the problem, DARPA is funding the infrastructure race, and companies like Pumpkinseed are building the picks and shovels. Syn Bio Beta this year, of course, focused on AI and bio, but it also gave me a lot of hope for some of the incredible work being done across the space, and more importantly, in the lab. That's your Deep Tech Brief for this week. I hope you like the deep dive on synthetic biology. I'm Max von Polnitz. See you next Thursday. Keep building. And as always, please comment and let me know what I should talk about next.